Okay, when we talk about DNA replication is the process of producing two identical replicas which is basically copies of DNA from one original DNA molecule. Now, when we talked about DNA is a double helix. You should know all the definitions of the double helix. To me, it's easier to turn DNA sideways to understand DNA replication. One of the things that has to happen during DNA replication is you have to unzip your double helix. Sometimes we use the word unwind. Sometimes we say it has to be unzipped. Either way, it has to be unwound and separated. So enzymes can synthesize, which means to make, new DNA strands. So, the way I like to do DNA replication is I like to draw it out. So, first, we're changing the DNA so that it's facing up and down. You can still say that these are the sugar phosphate backbones. You can still say that your nitrogen bases are going to point towards the inside. And you can still say that it's anti-parallel. Now, during the process of DNA replication, this double helix has to unzip. So, in order for replication to occur, first thing you have to do is you have to unwind the helix. So literally, you have an enzyme that we're going to learn that's called helicase. I'm just going to draw it right here. This enzyme is responsible for unzipping DNA. This is called helicase. Helicase, it's ends in ace. He's the enzyme that is going to unwind your double helix. Now, it looks a lot different. It's no longer double helix. You still have nitrogen bases. And when you unwind your DNA with helicase, you create something called the replication fork. This is the replication fork. It's basically the active portion of DNA where replication is going to occur. So I'm going to write that. You always draw your replication forks the same way. So, you have your replication fork. Helicase is the enzyme that unzipped it. So, helicase is the enzyme responsible for forming the replication fork. Now, something else has to occur in order for DNA replication to happen. An enzyme known as DNA polymerase has to come and build the new DNA. So you have an enzyme DNA polymerase that is going to attach to your DNA molecule and it's going to make a new strand of DNA. So, this is going to move in the direction as helicase, and it's going to take nucleotides that are complementary, and it's going to build a new strand. Now, DNA polymerase also is going to work in the opposite direction, and it builds a new DNA strand. So the new strand is going to be complementary to the old strands. So we say that basically during DNA replication, the two strands separate, and now the old strands are used as templates. So it is now a template strand. The old strand is a template strand so that the DNA polymerase can create a new one. So when we talk about DNA replication, I literally could give you an old strand and say that the old strand is AGG, CTA, CGA. So for replication, when you create a new strand, they have to be complementary. This is the template. The template is the old strand. So DNA polymerase is going to come build a new DNA strand, and you can link these together. So DNA polymerase during replication would say that A bonds with T, G bonds with C, so it's complementary to the template strand. C, G, A, T, G, C, T. So this would be the new strand that is complementary to the old strand, which is acting as the template. Now, that's DNA replication, but it has to occur on both sides. We call this the semi-conservative model, the way that we describe DNA replication. When we describe DNA replication, we say it is a semi-conservative model. You're going to keep one old strand and one new strand to create new DNA. So this is the replication fork. You create new strands on either side, and to describe that you're going to have one new strand. So in the end, when all this is said and done, you're going to have one DNA molecule that has the old strand, and one DNA strand that has the old strand, and you're going to have a new strand where the DNA polymerase actually bound it. And it's going to rewind, and this represents the semi-conservative model. So replication is when we create two new DNA molecules from one. But it's important to understand that this is the new strand on either side, and the template is the old strand. So we conserve half of our old or original DNA. So we have two new molecules. Now, when we talk about replication, we do call these daughter strands. Two new molecules of DNA are called the daughter strands. And we also would say they are semi-conservative or semi-conservative model. So I want to talk really quick about the semi-conservative model, and then we're going to end this until we start all the enzymes. So when you use the word semi-conservative model for DNA replication, this describes the mechanism of DNA replication. During DNA replication, two strands separate. That's the replication fork. Both strands are used as the templates. For free nucleotides to bind to create identical daughter strands. It is also important for the semi-conservative model that you say each new daughter strand has half of the DNA from the original strand that's the old strand, and half newly formed DNA. So you started out with your double helix. In replication, it has to be unzipped by the enzyme helix. New DNA is going to be formed by enzymes. And then you're going to end up with two new daughter strands that are half old and half new. You end up with two new copies. Half new formed, half old. This is the semi-conservative model. Now, this was discovered by two scientists known as Matthew Meselson and Franklin Stahl. And this happened in 1958. And they discovered the mechanism of DNA replication, and they called this the semi-conservative model.